So you can't talk about metabolism and exergonic and endergonic reactions without talking about ATP. And we've mentioned ATP before. It is kind of the unit of energy that life uses. It is one of the most common ways to store energy short term and use it to drive specific chemical reactions. So ATP is going to be our power source, our fuel for cellular work. And it's going to enable us to couple exergonic reactions that give off energy and to endergonic reactions, which need energy input. So that's going to be our intermediary. So a cell can do three kinds of work: chemical, transport, mechanical. But most energy is coupling in cells is mediated by ATP. So it's a very, very important molecule. What is ATP? Adenosine triphosphate. It's the cell's energy shuttle, right? So it's the same basic ATP that you would see when we build RNA, right? So if you look at the structure here, you see the ribose. It's ribose. It's got two hydroxyl groups on the 2' and the 3' carbon. So this is basically the same thing that you incorporate into a growing RNA strand, right? But we're going to chop off 2 of those phosphates to do that. Now, the key is that this molecule has a high free energy. It is very unstable, in essence. And because of that instability, we can harvest energy from it when we break the phosphate bonds on the tail of that molecule. So most commonly what we see is a release of energy when we go from a higher instability to a lower instability, when we go from the triphosphate to the diphosphate. So again, when you have a triphosphate, there is free energy there ready to be harvested. When I hydrolyze that phosphate group off the end, now I have inorganic phosphate and a diphosphate, adenosine diphosphate, and the difference then in the free energy between these two is the energy that we can harvest, right? That we can use potentially to do work. So what we're going to do is we're going to couple that to very specific chemical reactions, and oftentimes one of the ways that ATP does this is through phosphorylation. Just one example. So here in the synthesis of glutamine from glutamic acid and ammonia, we can see that the delta G for this overall reaction is positive. That means it's not going to occur spontaneously. You can put this in a jar, mix it up, and it'll never go forward unless you put energy into the system. So how are we going to do that with ATP? So ATP is going to hydrolyze that phosphate group, but in this case it's going to attach it to the glutamic acid. So now I have a phosphorylated glutamic acid plus ADP. Now that has a lot of free energy. Enough, in fact, that when I now combine the phosphorylated glutamic acid with the ammonia, I have a spontaneous reaction occurring. I'm releasing energy. So the net overall delta G of these combined coupled reactions is negative 3.9, and that negative sign is what you're looking for. The negative sign says that this can go forward. This is spontaneous, right? I have less energy or more stability in the products than I did in the reactants. In this case, though, I've just broken it down into 2 steps, right? So I've coupled 2 reactions to get an overall net negative delta G. All right. So again, this idea is phosphorylation. We're going to see phosphorylation again and again and again. We're going to see phosphorylation as an on and off switch with proteins. Okay, we're going to see all kinds of molecules that can be phosphorylated. It can be used from an energy point of view, but it can also be used from an activation point of view. Now, this idea of phosphorylation, we looked at that when we saw how ATP worked to pump things across the membrane, active transport, right? All of our motor proteins are going to burn, quote-unquote, ATP, burn through ATP to make ADP, right, to move down the cytoskeletal tract. Right? So again, go back and review some of the terms that we've talked about before in relation to ATP, and now hopefully it'll make more sense to you. So that review is very, very important. Now, the cool thing about the cell system is that though it is an open system, we can still recycle most of the physical stuff. Right? It's the energy that's moving through the system. So every time we split an ATP to an ADP, we've got a free inorganic phosphate out there waiting to be recoupled to an ADP to make ATP. So we're going to talk about how that happens. But we have energy, as long as we have energy coming into the system through the food that you eat, or in the case of a plant, the sunlight energy, we can continually generate ATP to be used and then regenerate ADP to ATP as long as we keep feeding energy into the system.